Okay. Now let's see the other type of secondary active transport, that is counter transport or antipode. So again you see we have shown the sodium potassium ATPase, which is maintaining the gradient for sodium ions from outside to inside the cell. And here we have shown a counter transport or antipode mechanism. What is happening, that it has a site for binding of sodium outside, right, while there is other substance, in this case calcium, which can bind on the inside, right. So calcium is moving from inside to outside from its low to high concentration, while sodium is moving from its high to low concentration from outside to inside. So why it is known as counter transport? Because one substance is moving from outside to inside while the other substance is moving from inside to outside, right. So that is why the name counter transport or antipode. But if you see, the fundamental functioning is same like that of co-transport, that is using the energy indirectly, and main energy is utilized by primary active transport for maintaining the gradient of sodium ions. So example of counter transport is sodium calcium antipode. So we'll just summarize a little bit. In active transport, we have seen primary active transport and secondary active transport. In primary active transport, we have seen that there is an ATPase which utilizes energy, and the substances always move from their low concentration to high concentration. While in secondary active transport, we saw that the energy is utilized indirectly, and one substance is moving along concentration gradient and the other substance is moving against concentration gradient. But depending on the direction of the movement, in secondary active transport, we have co-transport and counter transport mechanisms. Okay, now let's move on to third type of active transport mechanism, which is for large molecules, that is exocytosis and endocytosis. Fundamentally, in exocytosis, the substances which are targeted to move from inside the cell to outside the cell, they move by exocytosis. The substances are held in a vesicle, which is nothing but formed of membrane, and the vesicle moves to the membrane, that is the cell membrane, attaches to a particular site on cell membrane, and with this fusion, that site of the membrane breaks down and the vesicle causes release of the substance to the outside. On the other hand, in endocytosis, the substances are taken up from outside to inside the cell. So here you see the vesicle is forming from the cell membrane, and after taking of the substance, the pinching of the membrane occurs so that we have the substance in the vesicle within the cell. Now there are various types of endocytosis. There is a phagocytosis, pinocytosis, clathrin mediated endocytosis, caveola dependent uptake, and non-clathrin, non-caveolar mediated endocytosis. So let's see what are the different types of endocytosis and how they are operating. So first is phagocytosis. Phagocytosis is also known as cell eating. So large particles, dead tissue, they are taken up by the cell through this mechanism. So here the cell membrane is making contact with the solid particles, and then the cell membrane pinches off and forms a vesicle. One example of phagocytosis is being done by WBCs. Next is pinocytosis. Pinocytosis is also known as cell drinking. So here very small vesicles form, and the substances, very small substances are taken up in solution. So that is pinocytosis. Third one is receptor mediated endocytosis, what we called as caveola mediated or clathrin mediated endocytosis. So here is an example shown. So these are some proteins known as clathrin which are attached on the membrane. So these vesicles will form where these clathrin proteins are attached, and then there are certain receptors. As the ligands, the substances which need to be taken up, bind to the receptor, it starts forming a vesicle which is then pinched off from the cell membrane, and here that pinching off energy is used at the level of the pinching. So this is the coated vesicle or vacuole which is forming, and once it is formed, this clathrin gets separated from the vacuole and goes and attaches back to the membrane. So this is clathrin mediated endocytosis. So with this, we have covered all mechanisms of transport across cell membrane. Basically passive transport, which do not require biological energy, and the movement occurs along the concentration gradient of the substance and its types. Then the active transport, which requires biological energy and the substance moves against the concentration gradient, and the various types of the active transport. Thank you. Have a nice day.